Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So, in this connective tissue lecture series, I will be going over elastin and applied clinical aspects related with elastin. Let's get into the details. Elastin being present in extracellular matrix, it provides an extensibility and elastic recoil to tissues, and that is the speciality of elastin. And elastin is present in large quantities, especially in the lungs, large arterial blood vessels, and also in some elastic ligaments. And small quantities of elastin can be seen in skin, ear cartilage, and several other tissues. Like collagen, elastin has got a precursor. It will come from a precursor called tropoelastin molecule. And when it comes to the amino acid composition of elastin, 40% of elastin contains hydrophobic amino acids, meaning elastin is rich in hydrophobic amino acids. Elastin also contains hydroxyproline, but it do not contain hydroxylysine. Now, elastin is extensively cross-linked, and cross-linking of elastin, just like collagen, it needs an activity of copper-dependent lysyl oxidase enzyme, which will convert some of the lysine in tropoelastin into allysin residue. Now, one of the special feature of elastin here is formation of desmosine crosslinks, which is a tetrafunctional crosslink which is unique to elastin, and that is the 3 allysin links with 1 lysine within the tropoelastin molecule, and that is what is known as a desmosine crosslinks, and that's what is shown here in the figure. As you see, there are 3 allysins linked with 1 lysine, and this entire structure that you are seeing here is a desmosine tetrafunctional crosslink. Now, copper is required for this lysyl oxidase, which is involved in tetrafunctional crosslinks formation, which is unique to elastin. So, if there is a copper deficiency will lead to a dissecting aneurysm in the blood vessel, and that's one of the sign of copper deficiency among other signs of copper deficiency. And that sign of copper deficiency, which leads to dissecting aneurysm in the blood vessels, is because the copper deficiency will lead to defect in the desmosine crosslinks, because copper is essential for lysyl oxidase, which is participating in a desmosine crosslinks formation. That's how a dissecting aneurysm can be because of a copper deficiency, and that is related to desmosine crosslinks that is made by lysyl oxidase enzyme. Now, the main differences between collagen and elastin molecule. See, collagen has many different genetic types, whereas elastin is only one genetic type. Collagen has a triple helix, and such a triple helix is not found in elastin. Random coil conformation permitting the stretching process is found here, and the glycine XY repetition is there in collagen, whereas such glycine XY repetition is not seen in elastin molecule. Presence of hydroxylysine in collagen, whereas hydroxylysine is not there in elastin. Carbohydrate collagen is carbohydrate containing molecule, whereas elastin do not have the carbohydrate. And intramolecular aldol condensation is which leads to cross-linking in collagen, whereas intramolecular desmosine crosslinks are the ones that are present in elastin. And presence of extension peptides are there during biosynthesis of collagen, and such extension peptides are not found in elastin molecules. So, these are the main differences between collagen and elastin. Coming to a defect in or any mutations in a gene that is coding for elastin molecule, and one of the disorder here which is related to this is Williams syndrome, which is also known as Williams-Buehner syndrome. It is because of deletion in ELN gene, that is elastin gene, which is located on chromosome number 7q11.23. And this affects connective tissue and central nervous system. So, patients will show growth retardation, mild to moderate mental retardation, cardiac defect, specifically supravalvular aortic stenosis is seen. Abnormally increased levels of calcium in the blood occurs during infancy. That's what is referred as infantile hypercalcemia. And patients will show elfin-like facial features. Microcephaly, some facial features includes gargoylism, hypodontia, star-like or stellate pattern in the iris of the eyes are seen in Williams syndrome. Remember, this is because of mutation in a gene that is coding for elastin, that is ELN gene. Coming to a related disorder with elastin, and that is alpha-1 antitrypsin deficiency. Now the alpha-1 antitrypsin protein, it is synthesized mainly in the liver, and also it is synthesized from monocytes and alveolar macrophages. And alpha-1 antitrypsin, it inhibits number of proteolytic enzymes including elastase. And elastase enzyme, it degrades elastin. So alpha-1 antitrypsin, it protects elastin by inhibiting activity of elastase, and thereby alpha-1 antitrypsin, it protects our lungs and other elastin containing tissues from degradation because of the action of elastase. What if there is a mutation in a gene that is coding for alpha-1 antitrypsin? So mutation in a gene that is coding for alpha-1 antitrypsin may lead to emphysema and cirrhosis. That means it affects both lungs and also in the liver. So the type of mutation that you see in alpha-1 antitrypsin deficiency is a mutation in alpha-1 antitrypsin gene where GAG is replaced by AAG. That means G is replaced by A, guanine is replaced by adenine, which results in substitution of lysine for glutamic acid. And this type of mutation will lead to an abnormal form of alpha-1 antitrypsin, which will polymerize within the endoplasmic reticulum of hepatocytes, and it is not secreted into the blood. And that will give rise to severe deficiency of alpha-1 antitrypsin in the blood, and thereby its protective action on the lungs and other elastin containing tissues will be not there. That means there will be overactive elastase enzyme which will degrade elastic tissue, especially in the lungs. So that causes severe deficiency will lead to, severe alpha-1 antitrypsin deficiency will lead to a pan-acinar emphysema in adult life. Also note that cigarette smoking will greatly increase the risk of severe emphysema in these patients, because as such cigarette smoke has oxidants or free radicals which will oxidize a critical methionine residue in the active site of alpha-1 antitrypsin or available alpha-1 antitrypsin. The methionine is oxidized into its sulfoxide form, and the sulfoxide form of the methionine will decrease its affinity for elastase enzyme, which is a substrate for alpha-1 antitrypsin. So cigarette smoke will decrease alpha-1 antitrypsin activity by oxidizing a methionine which is present in the active site of alpha-1 antitrypsin. Thereby the binding of elastase to the active site of alpha-1 antitrypsin is decreased, and that will lead to overactivity of elastase enzyme and leading to signs and symptoms. So what are all the signs and symptoms we see here? Shortness of breath, wheezing or rhonchi, impaired liver functions can be seen, can lead to cirrhosis, and liver failure is seen in some patients. Treatment includes avoidance of damaging inhalants, intravenous infusion of alpha-1 antitrypsin protein can be done, and transplantation of liver and lungs is also considered. And also note that it is the leading cause of liver transplantation in newborns. So that is all about connective tissue, elastin and its applied clinical aspects. Thank you.